Hello and welcome to today's talk about next generation sequencing, which is also known as massively parallel or just deep sequencing. Next generation sequencing has completely revolutionized genome analysis. With next gen sequencing, it is possible to sequence an entire human genome in just one day. We start with the sample preparation. Once we have isolated our DNA, we need to generate smaller fragments. Now all of these DNA fragments will get ligated to certain oligonucleotides. There are two types here. These include sequencing binding sites and also an additional complementary sequence which can then hybridize to the flow cell. First, our DNA double-stranded fragment is denaturated. So in theory, the whole genome should be in fragments and all of the fragments have attached two different types of binding sites on each end. All the DNA fragments are now applied on a chip, the so-called flow cell. And as you can see here, there are two types of complementary sequences, the blue ones and the red ones in our example. The DNA fragments will at this ends hybridize with the flow cell as applied to the plate. To make this easier, we just look at one fragment. The first step here on the flow cell is the DNA amplification. By polymerase chain reaction, the complementary sequence to our single-stranded DNA fragment is synthesized. This process, which we have seen here, is done simultaneously for all DNA fragments on the whole flow cell. Now the DNA is denatured and the single strand, which is not attached to the flow cell's oligonucleotides, gets washed away. The next step is called bridge building. You may have noticed that we have two different types of oligonucleotides here, and the second type of oligo will now also hybridize to one of the sticky oligonucleotides on the plate. This process is called bridge building. The DNA is now amplified again, so a polymerase will synthesize the complementary sequence again. This whole process is called bridge amplification. The DNA is denatured, but now you can see that both fragments stick to the plate. The whole procedure will just repeat now. The DNA fragment will build a bridge again, and again we also have the bridge amplification. That whole process will be repeated for several rounds. And this here is the result. So out of one DNA fragment, we generated multiple copies by PCR. We have reverse and we have forward strands from our copy here. One of these strands here is cleaved now, and sequencing can begin. For sequencing, we just look at one copy now. As mentioned in the beginning, a primer can bind to the oligonucleotides. So this is a sequencing binding site here. Sequencing is now done with specific nucleotides. These nucleotides in this case are fluorescently labeled. Only one of them will fit. In this case, the T. The fluorescently labeled T will then be excited by a laser and the fluorescent signal is obtained. This is done with a color code, so we know that in this case blue is T. In the next case, the A will join. Again, the fluorescently labeled A is excited and we will receive a signal again, this time an A. This whole procedure is done for all of the nucleotides, one by one, and this is how we get the sequence. For simplification purposes, we just looked at one of these copies. Now notice that due to PCR we have several copies, so many light signals are obtained simultaneously. The last step now is the data analysis. So here informatic tools are required. The whole process of next-gen sequencing generated millions and billions of reads. These reads are now overlaid and compared to a reference genome. And by this overlay we can now generate the whole genome sequence. This was it. I thank you very much for watching. I put two interesting videos in the video description if you want to have further details about this topic. Otherwise, thanks and see you soon.